So our topic of discussion today will be the glial cells. Glial cells are, or the neuroglia cells are specialized cells that are present with the entire nervous system, along with the main neuron, the neuron which is responsible for conduction of signal. Along with it, all these different types of supporting cells that are present, and each and every one of them has got a specific function of their own, and with their help the total homeostasis or the proper functional condition of the neural system or the nervous system is what is maintained. Plus they help in the proper functioning, means the way the signal will be conducted, that particular function, and the way they can repair themselves or help in their existence or growth and development. Everything, it's all controlled by these different types of glial cells. Now the glial cells, they are mostly formed from the process of during the time of formation of the neuron. That very moment, the ectodermal region, the neuroectodermal cells or the tissues which are responsible for the growth or development of the neuron at the time of embryogenesis, that particular moment only all these cells starts arising, and these cells are the only one in the entire nervous system which are capable of mitotic division and continue its existence with numerous mitotic growth and all throughout the course of the life. And that particular situation, that particular growth condition is what is maintained or regulated, you can say. It's all by this neuroectodermal cells or the neuroectodermal stem cells that are still present in the body, but those cells are only capable of giving rise to this glial cells, not the neuron. So out here, this is the neuron, this structure. So I'm putting a little bit of shade to distinguish the neuron from the rest of the thing. So this is the neuron, and this neuron are supported by all these different types of glial cells. Let's begin with this one. So this glial cell is what is known as the ependymal cells. So ependymal cells are responsible for, let me arrange the entire pages properly. Yeah, so ependymal cells are responsible for like giving a little bit of protection or barrier to the entirety of the the nervous system tissue, the tissue which is mainly comprising the nervous system, that particular tissue, with that of the surrounding, we can say. And to be very specific, this is actually the one which forms the border between the the nervous tissues and the the cerebrospinal fluid. Actually, I use the word central because this can actually be the central canal of the spinal cord. We will be coming to that topic later on. So this, like the entire spinal cord, it has this small chamber running through it, which is the central cavity through which this cerebrospinal fluid is actually present. In case of the brain, it will be the meninges which has got the chambers through which the the cerebrospinal fluid is actually going through it. Okay, and this ependymal cells, it actually forms a barrier with of that of the nervous tissue with that of the surrounding, means with the meninges and all. So ependymal, its main purpose is production or giving protection to the this entire nervous tissues, and also it is responsible for the production of the cerebrospinal fluid. So that will be the major purpose of the ependymal cells. It comprises the entire barrier, total barrier or the covering of the central nervous system. It actually is made up of this ependymal cell. Meninges are separate. Now along with it, we will find next this particular type of cells which will be present at large. I just made one diagram to show. Now this I have made right here with the blood vessel. Okay, now blood vessel definitely will be there because blood vessel will supply nutrient and nourishment to every single type of body tissue. So brain is no exception. Brain will also receive nutrient and nourishment with the help of the blood cells, but this capillary is not going to like through this capillary as like the normal situation it occurs, where we find that the capillary, it actually produces means the juices, or what, I'm sorry, I used the wrong word here. Juices, not juices. It actually, to the capillary, all the tissue material means all the tissue material surrounding the capillary, they actually receive the nutrient and nourishment for their growth by means of the liquid that actually seeps out of the capillary, which is known as the lymph later on. It's being called the lymph or the interstitial fluid. So all this fluid or the lymph that actually comes out of this like capillaries will contain many things which are definitely necessary for the tissues surrounding it, but also will contain something that are not at all acceptable, not at all good for those cells. So all those material, all those liquid agents, all those chemical substances, if those chemical substances come and accumulate inside the neural tissue area, it is definitely going to be harmful for the neuron. So for that particular reason, these all liquids are not allowed to seep out that easily from this blood vessel. And to prevent that from happening, the brain actually creates a blood and brain barrier. Means blood and the brain barrier is created, and that creation of the barrier, it is mostly due to this particular type of cells. They are known as the astrocytes. Okay, astrocytes is actually a term which represent like astro means star like. Cytes is a word used, or cyte is a word used for representing cells. So it's a star shaped cells, and it appears like that. It has multiple projections running all around every direction, so that appears like a star like shape. Now astrocytes are not only present with this blood brain acting as a blood brain barrier, but along with it, it is present in the entirety of the neural tissue inside. Okay, so let's talk about the blood brain barrier here. So actually what happened is, I made only one like projections like this, but you can see I have coiled the entire thing. So one astrocyte will actually go and coil up the like the blood vessel, the capillaries. So once it is coiling up, so it will actually forms a kind of extension and projected layers around the capillary. Okay, and that will only allow the movement of the necessary particles, since water, minerals, and growth nutrient factors, everything, all those things it will allow to come out, but nothing other than that. Okay, because blood will contain even waste material flowing through it. So all those waste material also it will not allow to seep out. It will not allow to seep out any other unwanted chemicals, hormones, or whatever. It is not going to let any chemical come out of it. So it's going to be it's going to allow the selected material to come out from the blood to the neural tissue area. So that will be the purpose of astrocytes. Next, we have this particular cell. If you remember, we already actually discussed about it previous class. This is called the oligodendrocyte. So oligodendrocyte is responsible for formation of the myelin sheath. Okay, it is responsible for formation of this myelin sheath. So this myelin sheath that we see, it as you know, it's responsible for or it actually helps in the transmission of the signal through the neuron. Okay, so this myelin sheath is going to be very useful for the neural conduction, and this myelin sheath also gives a protection. So this oligodendrocyte is responsible for creation of the myelin sheath around all these particular cells. Okay, now along with it, we are going to find this type of cells are present there. These cells are known as the microglia. Okay, now microglia will be this cell and this cell, all of them. They are also present at large in the entire neural tissue area. So microglia is actually a cell which is not forming from the neurolectodermal tissue layer. The microglia cells are born from the hematopoietic cells which are which are responsible for the production of blood cells. Okay, so microglia has origin common to that of the blood. They all arise from the bone marrow region. They all arise from the stem cells of the bone marrow region, but they have the origin similar to that of the blood, whereas they all have common origin, means same origin as that of the neural tissue area, means the neurons and brain area. But these two area are actually, this type of cells are actually responsible, are having common origin to that of the blood. And their purpose is also as similar as that of the white blood cell of the blood. They are our immune cells of the nervous tissue. They have scavenging property just like the macrophages, and they go inside and find if anything which is unwanted, if anything that is harmful for the neural tissue that is present there, it will just simply swallow it and destroy it. So microglia are the immune cells of the entire nervous tissue, because blood immune cells will not be allowed to get inside this area, this particular area. It is only this one that actually majorly works there. Okay, now that comprises the entirety of all the cells that that can be found in the in this particular central nervous system region. But in the case of peripheral nervous system, there are small changes that occur. Okay, in peripheral nervous system, this oligodendrocyte will be replaced with Schwann cells. Okay, Schwann cells, if you remember, they are just like similar to that of that neuroglia tissue. Similar to that of the oligodendrocyte. Just it is a single cell that wraps around the entirety of the axon. If you remember, that is how it was represented, isn't it? It is one single cell that wraps around the axon, isn't it? Now if we want to elaborate that particular process, then we can actually make a small diagrammatic representation of how it actually happens. So we have the axon. Okay, so we are going to find that this particular neuroglia, the Schwann cell, so the Schwann cell will come here and will actually wrap itself around the initially to the wrapping will not be completely permanent, so there will be gaps. So it will wrap itself around this the axon region. Okay, first it will come and just simply try and grab it. The next thing that it will do means out from here, the next step that will occur will be that the wrapping is now getting a bit intensified, we can say. Okay, so it will actually intensify itself, means it is actually now going round and round and started forming the sheaths of myelin inside. Okay, so finally we are going to find that it is actually coiling up the entire axon. It has coiled up the entire axon, and we'll find numerous layers of this myelin sheath around the entire of the axon, and for that it actually just simply wraps itself around, means it just comes, grabs it, and then simply coils the entire thing around with the myelin sheath. Okay, that is how the Schwann cell actually occupy or grab around the entire we can say the entire axon. Okay,